In this video, we will discuss about virus, one of the topic of virus cycle, that is uncoating. So, in the previous video, we have discussed different kinds of penetration step. One of them is direct, and another one is endocytosis mediated penetration. In this endocytosis mediated penetration, we have discussed about two types. One is for the enveloped protein, enveloped virus, and another one is for the naked virus, that is only with nucleocapsid. Now we will discuss about uncoating. And uncoating is also divided into two parts mainly. One is cytoplasmic trafficking, another is nucleus trafficking. So what is cytoplasmic trafficking? When the genetic material of the virus is replicated and expressed in the cytoplasm, then the virus need to be exposed, or virus need to be available in the for the cell machinery in the cytoplasm. When this is usually occurs in the case of RNA, as RNA is translate most of the time in the cytoplasm. So let us consider an example. Consider this as the nucleocapsid, and this as the envelope. And suppose it is entering with the help of endocytosis mediated penetration. And these, there are microtubules that are present, and with the help of microtubules motor, these are microtubules, and with the help of microtubules motors, this vesicle containing virus can be transported to the other parts of the cells. Usually it has two ends, microtubules. One is positive and there is negative. Negative is more close towards nucleus, and positive is more close towards cell membrane. So what happens in this is that there are microtubules motors, you can say, that carry this vesicle containing virus to the negative part. And there are two general motors. One is dyne. Dyne usually carries, dyne is the microtubule motors that usually carry vesicle proteins from positive to negative, and there is another microtubules motor that is kinases. The spelling might be wrong, but you can check them in the reference paper that I have provided you in the description. This kinases helps in transferring the intracellular parts from negative to positive. So this is also a microtubules motor. So what happens in case of cytoplasmic trafficking This dyne helps in transport of this vesicle protein from cell membrane towards inside the nucleus, inside the cells within the cytoplasm, where most of the cell machinery that is responsible for replicating and expressing this genetic information present within the virus are available in the cell membrane. These are usually present near the nucleus, so this microtubule helps in transferring the vesicles near the nucleus. And this happens when the enveloped, this case that I have described is about the virus that is enveloped, and if the virus is not enveloped, then case also remains same. You can see that in case of non-enveloped virus, a nucleocapsid interact with this microtubules motor. Nucleocapsid directly interacts with microtubules motor and helps in the transfer of genetic material from near the cell membrane towards the nucleus. And there is another case that I have already discussed, or I have already informed you about in the penetration stage. There are some nucleocapsids and some nucleocapsids that have the some component within it that can lyse this the coating or the vesicle that is made by plasma membrane, and then come out into the cytoplasm for replication and expression. So this I have talked to you about cytoplasmic trafficking. Now we will quickly go through how the genetic material is transferred to nucleus in case the genetic material is usually transcribed or translated in nucleus. In case of nucleus trafficking is generally happen in a case when the genetic material is present in the form of DNA. So suppose this is the nucleus membrane, and when the genetic material is very small, like in the case of polyoma virus, there are pores in the nucleus membrane at certain point of time, and if the nucleocapsid is very small, if the genetic information or the genetic material of the virus is very small, such as in the case of polyoma virus, then it can directly pass through the nucleus membrane and can directly pass through the nucleus membrane, and the genetic information can be transcribed and translate with the help of cell machinery. But if the genetic information is big, such as in the case of adenovirus, in adenovirus, the capsid cannot go through the plasma membrane because its size is bigger than the pore of the nucleus membrane. So in that case, it will dock with the nucleus membrane, and during this docking process, there is some misalignment or slightly dislocation of the nucleocapsid occurs, and through this through this step, it will allow its genetic material to move inside the nucleus lumen. And similarly, the case occurs with herpes virus. In case of herpes virus also, the genetic information is quite big, and it will not be able to pass through the nucleus. So in that case also, the nucleocapsid of the herpes virus makes a dock with the pores of the nucleus membrane, and in this case the disruption happens is low compared to the adenovirus. However, both of them does not affect much, as the genetic material that they need to transfer it into the ER lumen will occur without having its impact on without having its impact on the structure of the nucleocapsid, whether the nucleocapsid is too much disrupted or less disrupted. The genetic material will pass on to the nucleus, within the nucleus, once it docks with the nucleus pore. This is how uncoating of the genetic material occurs in the cell membrane, and now the cell machine occurs in the cell, not in the cell membrane, and now the genetic material is exposed to the cell machinery for expression and replication. So this is all for this video. Thank you.